Hello, everyone, and welcome to the DocTIS project webinar, Discovery of Combination Therapies for Immune-Mediated Inflammatory Diseases. We're thrilled to have you all here. Thank you very much for taking the time to be part of this session. We hope we can make this a valuable experience for you. The DocTIS project is a European Union-funded initiative that focuses on identifying more effective therapies for immune-mediated inflammatory diseases, specifically ulcerative colitis, Crohn's disease, rheumatoid arthritis, systemic lupus erythematosus, psoriasis, and psoriatic arthritis. By leveraging well-characterized IMID patients' data, high-quality biological samples, multi-omics data generation, and systems biology approaches, the project aims to discover combinations of targeted existing drugs that are more efficacious at blocking chronic inflammation than the current single drug practice. The project consortium is composed by the Vall Institute de Recerca, the VHIR, who also acts as coordinator, Cardiff University, Verona University, Charité Medical University, IDIBAPS, Centro Nacional de Análisis Genómico, CNAG, Immune-Mediated Hudson Alpha, and Zavala Innovation. To provide a deeper understanding of the DocTIS project, we have with us Dr. Sara Marsal, coordinator of the project and head of rheumatology department at Vall Barcelona Hospital Campus, Maria López Lasanta, postdoc rheumatology researcher at the Vall Barcelona Hospital Campus, Dr. Holger Heyn, single-cell genomics group leader at Centro Nacional de Análisis Genómico, CNAG, Dr. Antonio Julià, head of rheumatology research group at Vall Barcelona Hospital Campus, Dr. Pere Santamaria, pathogenesis and treatment of autoimmunity research group leader at IDIBAPS, and Dr. Ernest Choy, professor and head of rheumatology department at Cardiff University. After all the speakers have participated, we will begin the question and answer session. You can write your questions in the chat at any time, but we kindly ask you to specify who your question is addressed to. This will help us ensure that the right speaker provides the best response. We start the session with Dr. Sara Marsal, coordinator of the project and head of rheumatology department at Vall Barcelona Hospital Campus, who will do a general introduction of the DocTIS project.
Thank you very much, Mariano. So hello everyone. We are going now to start the webinar on DocTIS project. I would like to thank you for all of you to be here today. It is great to welcome you. Just one moment, please. It is great to welcome to all of you to our 5 years General Assembly of the DocTIS project. So as you can see here in this first slides, the title of the project and our aim is to identify optimal combinatorial therapies in immune-mediated inflammatory diseases using system approach. Happy to tell you today and tomorrow, all the partners of the DocTIS consortium, we met here in Barcelona, all of us, talking about what we have done in the past year, talking about what we have learned about these diseases, and just to better define what we have to do this last year of the project to reach the final objectives of this project. As you have seen, this project is about IMID diseases. So these are immune-mediated inflammatory disease, which is the main focus, the focus of our research in patients. First of all, let me introduce a little bit the project and also the partners, the DocTIS consortium. To the next slide. Okay, here in these slides, you can see the DocTIS consortium, the partners. So the DocTIS consortium is a biomedical research partners with 5, that includes 5 different European countries and also United States. As Mariano said at the beginning, so Vall the partners are Vall Research Institute, which is led by myself, Sara Marsal, and also Dr. Julià. Cardiff Institute, Ernest Choy, Verona University, led by Giampiero Girolomoni, University of Charity, led by Britta Sigmund in Berlin, IDIBAPS here in Barcelona as well, Dr. Pere Santamaria, CNAG also here in Barcelona, Dr. Holger Heyn, IMID-omics, Manuel López, leading IMID-omics in US and also here in Barcelona, and Hudson Alpha Institute for Biotechnology, led by Rick Myers. So I would like to give my deep and sincere gratitude to all the partners for their teamwork and dedication to the project. With such a strong project support to the project, so we are able to make a big progress. As I told you at the beginning, we are focusing to study IMID diseases. What are the IMID diseases? Let's talk a little bit about this disease. IMID diseases are the disorders characterized by chronic inflammation resulting from dysregulation of the immune system that affects different parts of the body, different organs or different tissues. They are quite common diseases with a high prevalence, about 7% of the general population, and these patients needs medical attention from different clinical specialties, rheumatology, dermatology, and gastroenterology. Unfortunately, despite it has been great advance in the last 2 decades in the treatment of these diseases, so unfortunately, there is no cure so far for these diseases, and all the patients need lifelong treatments. One examples of these diseases that we are working in these diseases are rheumatoid arthritis, which is a polyarthritis, psoriasis, which is a skin disease, psoriatic arthritis, which is the psoriasis, the skin disease plus articular inflammation component, 2 more prevalent types of inflammatory bowel disease, which are ulcerative colitis and Crohn disease, and also systemic lupus erythematosus. What is these patients are treated so far? The current therapies in most of these cases are immunomodulatory targeted therapies that are used to reduce the inflammation in the different parts of the body. These therapies mainly target, in most of the cases, block different proteins that are associated with the development of the disease. However, as I said before, beside the great advance in the last 2 decades, these therapies are costly, and as this disease affects millions of people during their working life, so this represents a high burden of cost for the medical care, even at the same level of the cancer care. Another issue is that most of these treatments, they are quite efficacious, but there are some patients that do not respond to these therapies, and between the patients that do respond, over the time of the therapy, they used to lose the efficacy of these treatments. And also another important challenge is that at the level of the physicians, we do not have any decision-making tool that help us to choose the best treatment for each patient. So the DocTIS project aims to face all these challenges to better identify new treatment strategies for these patients. As I said, the DocTIS objective is to identify therapeutic combinations that are much more efficacious and safer than the current single drug approach by using system biology. This objective is based in 3 main statements. All these diseases, many of these diseases, share a biological future. So this is something that is already new. So many developed therapies for one IMID can be beneficious for other IMIDs. Also, we know that, and we believe, and we know for another therapeutical areas that combining treatments, we may be able to reduce the drug dosage. So in that case, we can combine the drugs with lower doses, we will be able to minimize the side effects associated with high concentrations of a single treatment. And also important is that combining these drugs that already are in the market, we could enhance the effect of each one, reducing the probability of becoming resistant to the therapy, which make that treatment that is efficacious at the beginning, over the years will lose the efficacy. So with the aim to bring these new strategies, we come with this project with this specific design. The DocTIS project is based in a longitudinal study using modern high-throughput technologies on samples from very well-characterized IMID patients. The way that these huge projects work is structured in work package. So work package 3, for example, is the patient selections in where we select those patients under a clinical evaluation. In that case, we have studied the 6 IMIDs that I have told you, where these patients have been treated with 6 of the current approved drugs. And we have classified these patients in very well responders and non-responders to study the difference among them. Work Package 2 is a high throughput data management to handle the large volume of data that has been generated in this project. Work Package 4 is the molecular data generation. So from samples obtained from these patients, responders and non-responders, we have generated a large molecular information with a different source of molecular information. We have analyzed the genotype, which means all the genomes that a person could have, Vall transcriptomic, which represent the expression of all the genes that a given person could have, single cell transcriptomes, which means the expression of all the genes in a single cell, in a unique cell, and the proteoma, which means to analyze all the proteins that are circulating in the blood in a patient in a given time. So then, with all this information, clinical information and molecular information, we move to Work Package 5. In this work package, system biology analysis, we have developed a computational methodology to identify, to select the optimal drug combinations from these IMIDs. So with this methodology, we have been successful to identify the optimal drug pairs that to be tested in the patients. But also, we want to go further, and we would like to identify personalized biomarker therapy to move for precision medicine. So it's our aim in Work Package 7 to design a low-cost and informative methods to predict the clinical response on those patients that will be treated with the combinatorial therapy. Then the Work Package 6, what we pretend, our aim in this Work Package 6 is to develop information from a preclinical validation of this combinatorial therapy. What are we doing here? So we evaluate the combinatorial therapy that we have, the combinatorial therapies in different models of this disease, in different animal models of this disease. So in that case, we compare the therapeutic efficacy and the potential toxicity of the combinatorial therapies versus each of one monotherapies in animal models. So this gives us confidence about the efficacy and toxicity in animal models of these diseases before to test these in patients. Work Package 8, which is more important, is the large big work package. We will perform a clinical study validation of the chosen combinatorial therapy. This clinical trial represents proof of principal validation of the efficacy of the combinatorial therapy that has been identified with our methodology and with the information with the animal model in different patients. So this will provide us preliminary data about the safety and tolerability on the selected combinatorial therapy. And final, very briefly, but not less important, I would like to mention two work packages, Work Package 9 and Work Package 10. Work Package 9 is about dissemination and exploitation because it's our aim to share with all the scientific community, with medical community, and with all the population to share our methodology and the results of the study. And Work Package 10, which is related with the ethics requirements. In this case, we want to ensure that all the steps that are performed, protocols, steps, all the analysis that we are doing in this project fits the strict ethical requirements to protect the patients and to be completely aligned with the international standards. So finally, this is the last slide. I would like to thank you again, all the partners that are contributing to this project. Without this team effort, this would not have been possible. So working all together, I would like to tell you that we are almost closer to bring new strategies, new therapeutic strategies for these patients to improve the quality of life. I would like to say thank you also the European Commission to invest in the improve the knowledge and the therapeutic strategies in this disease, in this chronic and very disabling disease, the Immune-Mediated Inflammatory Diseases. And also, I would like to thank you all the patients that some time ago have provided samples to contribute to the investigation in these diseases, which has been extremely valuable for the samples. So if you would like to learn more about the DocTIS project, you could see our webpage, doctis.eu, and have to tell you, as I said, Maria, at the beginning, that we are very active in the social media, so we could follow us in the LinkedIn or in the former Twitter, in case you would like to follow our results and our dissemination and communication process. So thank you, everybody, all the partners, and thank you to stay, all of you here today. So I'm going to turn off Marianne, right?
Okay, so thank you, Sara. Next presenter will be Dr. Maria López Lasanta, postdoc rheumatology researcher at Vall d'Hebron, Barcelona Hospital Campus, who will explain the clinical features of patients at the IMID Biobank in Work Package 3 patient selection.
Good afternoon. I'm going to present the Work Package 3 of the DocTIS project. We will see an overview of the design of the study and how patients were selected. The objective of this work package are to determine the screen response feature for the 11 immune-mediated inflammatory disease of that combination based on the IMID biobank cohort, and to select a good responder and co-responder for each drug per IMID, more than 150 patients to 2 time points that will be analyzed using high-throughput technologies in Work Package 4. The design of the study is summarized in this slide. Let's go through each of these 4 graphics individually to learn about the details. First, patients were recruited across the center of the IMID consortium. The IMID consortium is a network of researchers from all over Spain, founded by Dr. Sara Marsal. It's allowed to recruit IMID patients and their clinical data using homogeneous and standardized protocols. IMID biobank of the Vall Institute Hospital is where samples collected were stored. The IMID biobank complies every year with ISO certificates, a standardized procedure, and has more than 17 years of experience in securing and controlling the sample storage condition of biological samples from IMID patients. Also, it's very important that Vall Research Institute is one of the participant institutions in this process, being the coordinator of the sample collection process. For this study, patients starting a targeted therapy were recruited. At the first visit, physician complete clinical questionnaire and patient samples are collected. In addition to the first visit, at least a second visit is planned. Importantly, having clinical and molecular data simultaneously is key to characterize the biology of the response to the treatment that these patients are undergoing. Now we move to the eligibility criteria. Next, the clinical experts of the DocTIS consortium, which are from Cardiff University and Vall Research Institute for Rheumatological Diseases, from Charité University Hospital in Berlin for gastroenterology, and from University of Verona for dermatology. They decide patient eligibility criteria. This includes strict diagnosis criteria endorsed by the scientific community. XLR criteria in the case of rheumatoid arthritis. Also, patients were recruited not to have relevant comorbidity and have a well-defined response to the therapy. Regarding the IMIDs included in this study, we have patients with rheumatoid arthritis, that is a chronic autoimmune disease causing joint inflammation, pain, and structural damage. We select patients with rheumatoid arthritis treated with anti-TNF therapy, anti-AL6, and JAK inhibitors. Then we have a patient with psoriatic arthritis, that is an inflammatory arthritis associated with psoriasis. And we include patients treated with IL-12 and 23 and anti-AL-17. Then we have patients with SLE, that is a systemic autoimmune disease affecting multiple organs with period of flare and remission. And we include patients treated with belimumab, with anti-VHIR. Also, we have patients with psoriasis, that is a chronic immune-mediated skin disorder, and we select with the patient treated with anti-EL-17 and anti-TNF therapy. Then we have patients with ulcerative colitis, that is a chronic inflammatory bowel disease causing inflammation of the colon and rectum. And we select with patients with anti-TNF therapy. And finally, patients with Crohn disease, that is a chronic inflammatory bowel disease that can affect any part of the gastrointestinal tract. And patient treated with anti-TNF therapy. Finally, also healthy volunteers were recruited to have a good control. Since all these diseases are IMIDs, they share biological similarities. So treatment that are not approved for one of them will still work in another. In each of these controls, patients were undergoing one of these therapies. But later, in analytical phase, the molecular information will be crossed between groups to evaluate any potential combination. Once the eligibility and the groups were established, patients were finally selected. 8 responder and no responder were chosen for each IMID therapy combination. In this stage, the bioinformatics team from IMID collaborated with the clinical expert from DocTIS to find the best possible selection. Patients were chosen not only to be very well responder or non-responder, importantly, in order to obtain high-quality statistical results, responder and non-responder have to be balanced with respect to the potential confounders, like sex, age, or baseline activity. The final stage is the generation of molecular data. In total, after selecting the patient from cohort, more than 170 patients and 2 time points and 8 healthy controls were chosen to generate molecular data. The samples were randomized by the IMID bioinformatics team and were sent to the corresponding data generating institution, Hudson Alpha for genotyping, and transcriptomics, and CNAT for single cell transcriptomics. So that is, thank you all for your attention.
Thank you, Maria. Now it's time to hear from Dr. Holger Heyn, Single Cell Genomics Group Leader at Centro Nacional de Análisis Genómico, NAG, and leader of Work Package 4, Molecular Data Generation from IMID Biobank Patients.
Good afternoon, everyone. Thanks, Mariano, for the introduction. I will mainly talk about Work Package 4 and I will mainly talk about single cell genomics because that's what my group has been contributing. Also, that is what we have been specialized on over the last almost a decade now. Initially, as a group, we have been working mainly on the technical side, kind of scouting technologies, looking at how they benchmark against existing technologies, defining standards. If needed, we develop computational tools, but always with the vision to apply these single-cell resolve technologies in the human context. And here we're working on 2 scenarios. One is the Human Cell Atlas project, which is a project to digitalize the health of the human. But then eventually we want to apply these technologies in the clinical context, and that's why I'm very grateful to our partners at DocTIS for this opportunity to work on inflammatory diseases. Why doing single-cell genomics? I think that's kind of the elephant in the room. Because bulk sequencing, so looking at tissues as a whole is much cheaper. So why do we make the effort looking at single-cell levels? And you can envision that looking at a lung as an organ or skin as a tissue, that these tissues are not kind of composed of the same cell types or cell states, but it's kind of a mixture of different cell identities. And that's where single-cell technologies shine. So the tissue is not mixed up. We look at the average of it, for example, transcriptome profile, but we look at individual cells. And with this slide, I will walk you quickly through the process, how we generate these single-cell resolved transcriptome profiles. So we would start with cells in solution. So we take a lung or a skin tissue, dissociate single cells, and then we would encapsulate those single cells into single droplets. And within those droplets, we assign an index to the individual cells that then allows us to pool those cells together and basically digitalize their molecular context. In this case, we're looking at messenger mRNA molecules. And in practice, it looks like this. So we have many of these droplets generated, around 100,000 to 200,000 droplets. And then we want to make sure that each of the droplets contains a single cell. So those droplets here highlighted with the red stars. And we want to make sure that we have the minimum amount of empty droplets and the minimum amount of 2 cells within one droplet. So this is kind of the target, how to generate high quality single cell data sets, how high quality single cell transcriptomes. On the next step, then we would digitalize those cellular profiles. So each of these kind of quadrants here would be reflecting the single cell profile. So each individual cell would be characterized by a single cell transcriptome. And we use that then in the next step to assign similarities of cells. So in this case, we are looking at our initiative to generate the human tonsil atlas. So these are only immune cells from the healthy human tonsil, part of the Human Cell Atlas project. And here in these so-called UMAPs, each of the dots would represent a single cell. And the proximity of those single cells then allows us to define what is the complexity of immune cells in this case, the human tonsil. So here, then the clusters color-coded in the different colors. On the left-hand side, the B cells, on the right-hand side, the T cells, really allows us to separate major cell types. But it also allows us then to zoom into the different compartments. So on the left, you see the kind of overall composition of the tonsil with the major cell types being present. But then we can zoom into each of the cell types and each of the cell lineages, really to define a fine-grained map of cell types and states forming the human tonsil. In this case, we identified 121 different cell types and states using our tonsil Immune Cell Atlas project. So this is kind of a representation of the T cell compartment. So here we are zooming into the T cell compartment in detail. And again, we could look at the similarities and heterogeneities across the entirety of T cells in human tonsil and define lineage commitment, so from early naive activated cells to later differentiated cytotoxic or regulatory T cells. So in general, what single cell sequencing allows us really to define the very high granularity, heterogeneity we have within a sample. And this then we use in the context of Immune-Mediated, in the context of DocTIS, really to look at T cell cells in circulation and then to compare healthy against the disease context. For projects like the Human Cell Atlas project, we then not only look at a single organ, but multiple organs, and then define the cellular resolve maps, organ by organ, tissue by tissue, and then put this together in the full kind of digitalized healthy human blueprint that, for example, in DocTIS we used as a blueprint for the healthy background, and then really to define what changes we identify in diseases. What we've done specifically in the DocTIS consortium is we looked at a large number of different inflammatory diseases, and we brought this into the context of even more chronic and acute diseases. So in total, we looked at more than 6 million cells, 20 different conditions, chronic or acute inflammatory diseases. And again, on the right-hand side, you see these UMAPs So a massive amount of single cell now grouped by cell types and states, but also you can see already differences between the different diseases. And then our approach was to look at the different inflammatory profiles, to look more into the mode of action and the origin of diseases, potential druggable targets, but we also had initiatives to identify biomarkers, for example, to diagnose or to predict disease outcome. But for me, one of the most important outcomes was our goal to classify patients and diseases based on cells in circulation. So here within DocTIS, we're looking at PBMC samples, so immune cells from the blood. And the goal was then to use a blood sample from the patient to classify precisely what disease that patient is suffering from. And here, you don't have to kind of look at the details, but you just look at the color code where on the left-hand side, we're classifying our patients that were not in our trainings. So these are patients that we newly classified based on our DocTIS references. And you see based on the kind of a yellow color code on the left-hand side that we could very precisely diagnose the disease based on immune cells in circulation. However, then we increased the complexity of our scenarios. We were completely taking studies outside from the DocTIS in our training set, and this ended up with a quite poor classification as seen in scenario 3 on the right-hand side. However, then we took a step back and only used the DocTIS set to remove complexity, such as data being being generated with different centers. And this really allowed us to some extent, really to use immune cells in circulation and to look at the different cell types and state that we classified initially and quantify their potential to serve as a diagnostic tool, using cells in circulation to classify a patient to a given disease. And this for us is the first step towards a universal classifier, where then single cell technology are used in the clinical context and are used as a clinical diagnostic tool to precisely quantify and identify what a patient is suffering from. And with this, I will hand over to the next speakers. Thank you all for your attention. And over to you, Maria.
Okay. Thank you, Holger. We continue with Dr. Antonio Julià, Head of Rheumatology Research Group at the Vall d'Hebron Barcelona Hospital Campus, who will explain Work Package 5, Systems Biology Analysis.
Good evening. Thank you for attending this online presentation from DocTIS. So, as it was mentioned, this European call objective was to develop systems biology approaches for the identification of drug combinations in immune-mediated inflammatory diseases. So, first of all, we need to understand, have a global overview of the type of system that we are working with. The immune system is a special type of system that defends us against microbes, foreign substances, damaged and malignant cells. And compared to other organs and systems in the body, it has some peculiarities. It's dynamically distributed, and it's a network of cells and molecules that together work to safeguard us from external pathogens and cancer cells, for example. But in immune-mediated inflammatory diseases, this system works against the cell with a genetic susceptibility background that we have advanced a lot in the last 15 years, and unknown environmental triggers. This leads to the development of an autoimmune reaction against cells and tissues of the individual. For example, the joint in rheumatoid arthritis or the skin in psoriasis. A common thing is that to date, we don't have a cure for this disease, and therefore the medical health challenge that we have right now is to find therapies that can reduce at least the activation of the autoimmune response and leave patients with a good quality of life. So how have we done this so far? First, like in the mid-20th century, the first immunosuppressants were developed. immunosuppressants are kind of a misnomer because you don't want to stop the immune system from working because that would lead to infections or to cancer. You want to reduce its activity to, in this case, reduce chronic inflammation in IMIDs. The problem with this first type of drugs is that they had a broad immune modulation. So they affected many other cells than the ones you were interested in. So you have to be very careful with the risk, avoiding toxicity and secondary effects. And most of all, they were not very efficacious. And so there were thousands of patients that were still having very crippling diseases. Fortunately, 25 years ago, then a new era of therapeutics came into, especially started by anti-TNF therapies, in which by knowing the precise mechanisms of the immune system and attacking specific parts of this system, we are able to do a very specific immune modulation. This way, we can have much more efficacious therapeutics, and we can reduce secondary effects like toxicity or other type of problems. These types of therapies that are so specific in what they are targeting, they have shown to be very efficacious in a subset of patients. And this is important, and it's what has brought us here. Because there's also a large portion of patients who do not benefit, or even in some cases, some patients that start responding well, they eventually lose efficacy. So there is a big health problem that affects millions of people in Europe and in the world. So the objective of our project, the DocTIS, is centered into tackling this problem. Now, how can we improve the current therapeutics ceiling that we have right now, that we cannot provide our patients with a sustained clinical remission? That's what we want, no symptoms of those patients. So here we have been developing a systems biology approach to, by using the data that has been previously explained by Dr. Sara Marsal and Holger Heyn. We are using this type of data to try to find what would be the right pair of drugs that we should use to improve this therapeutic scene. So for this, what we are using in the rheumatology research lab in collaboration with IMID, two of the partners of DocTIS, is a strategy that is based in a principle called signature reverse principle. This principle has been used for a while in the search for drugs in cancer, and what we have done here is an adaptation of this. First, explain you a little bit what is this principle. Basically, you get samples from a patient and from a healthy individual, in this case a cancer, and a healthy individual, and you extract molecular data. From here, you obtain a signature of disease by comparing what are the levels, for example, of RNA in a set of samples and the levels of RNA in cancer. Next, what you do is you profile hundreds or thousands of compounds in vitro. You treat cells with different compounds and see how they respond to this therapy. Finally, the last step is you merge these two signatures, the signature of disease, you contrast it against the signature of the therapy, and see which one of these therapies is better at returning the signature back to the healthy states, to the homeostasis. So, as I said, this is a technique that has been used for a long time in cancer because it's amenable to that. You can put cancer biopsies in vitro and test a lot, but immune-mediated inflammatory diseases, you cannot mimic that because it's a systemic disease, as I was commencing to explain at the beginning, and so we have to look for an alternative. And the alternative that we are looking at is an approach that we, at the Rheumatology Research Lab at Vall, in collaboration with IMID, we underwent during the COVID-19 severity crisis. Like many research groups around the world, we tried to put a little grain of sand to help in this major problem, to avoid severity, in COVID. And here, what we proposed is on this signature-based principle to use, in this case, blood, to use blood of COVID-19 patients with severe disease and healthy individuals to take this disease signature. And then instead of analyzing in vitro signatures, what we did is obtain signatures of response in vivo. That is, analyzing the gene expression of a patient that receives the therapy at week 0, and then several weeks later, see what are the molecular changes throughout all this time. So with this adaptation, what we were able to find is that abatacept, a drug that blocks CD8, the CD86 pro-inflammatory mechanism, was very promising, very strongly associated to the reversion of this signature induced by COVID-19. This was, we published this, and we were very happy to see that months later, the NIH started a clinical trial called Active-1, in which among 150 different therapies that they considered to include in this clinical trial for COVID-19 severity, one of the 3 drugs that was included was abatacept. We were even more happy to hear that at the end of this clinical trial, it was observed that abatacept was clearly associated to a reduction of the odds of death, and also it increased the probability of improvement in patients, thereby supporting that not only abatacept is good for COVID-19, but this strategy could be used for this type of diseases that have a more systemic component like COVID-19, but in our case, of IMIDs. So this is the strategy that we are proposing for this problem, the system biology approach. So this is the objective that we have at hand. We want to develop a systems biology approach that can leverage the molecular data that we generate from the selected patients that Maria commented previously, work package 3, work package 4, so that we're able to identify the best combinations that we can use to specifically treat each IMID. This, as will be explained later, is being validated in animal models, and eventually this is being tested in patients to corroborate these predictions. Thank you very much for your attention.
Hey, thank you very much, Antonio. Next presenter will be Dr. Pere Santamaria, Pathogenesis and Treatment of Autoimmunity Research Group Leader at IDIBAPS, and also leader of Work Package 6, Preclinical Validation of Combinational Therapies, who is joining from Canada.
Please, next slide. Okay, so in this work package, what we're trying to do is compare the therapeutic efficacy of specific biologics, the drugs that have been discussed in the previous talks, either in isolation or in combination in different mouse models or preclinical models of chronic immune-mediated inflammatory diseases. So rheumatoid arthritis that affects the joints, psoriasis that affects the skin or the joints as well as other organs, or inflammatory bowel disease where there is inflammation of the terminal or distal part of the small gut, a disease called Crohn's disease, or the large gut, which is referred to as ulcerative colitis. And so we have animal models that are spontaneously, or types of animals, mice in this case, strains of mice that specifically develop spontaneously some of these diseases, or where one can induce those specific diseases. And then we use these animal models once they are sick to see how they respond to different treatments. Next slide. So I'm just going to briefly go through a few slides on different diseases. So this is for psoriasis. We use drugs that are chosen, by our colleagues in the consortium that are known to have, that are approved for the treatment of the disease, and that are expected to perhaps work better in combination than in isolation. So we try the monotherapy, so these two particular drugs, ustekinumab and secukinumab, and we compare their therapeutic efficacy as compared to a control drug. A control drug is something that is biologically similar but does not have biological activity. So in gray shows the lack of therapeutic efficacy of the control. So the higher the bar, the higher the histogram, the worse the disease is in the animals. And then in blue and in red are the effect of the monotherapies. You know, blue is one drug, red is another drug, and you can see that the size of the bars decreases in these animals, meaning that the animals are less sick. Means that the drug is working at reducing disease in these animals. When you combine these drugs together, and now I'm just showing in purple the therapeutic efficacy of the combination of these two drugs as compared to their individual treatments, blue or red, you can see that there is improved therapeutic efficacy, right? So next slide. So in psoriasis, then, combinations of effective drugs improve therapeutic outcome relative to the individual drugs, even at low doses. I haven't shown you this, but we've tried lower doses as well as higher doses, and lower doses work just as well as the conventional, approved doses. So this suggests that low doses of specific drug combinations might yield similar therapeutic effects than high doses of the corresponding monotherapy. So combinatorial therapy may allow one to reduce the amount of drug that is delivered in combination and obtain the similar therapeutic effect. Next slide. So that's one of the lessons of that particular experiment. So this is in rheumatoid arthritis. Here I'm just showing you 3 different drugs. At the top, we just compare the extent of swelling in the joints of the animal. The higher the value means the more swollen the joints and the more joints affected are in the animal. So it means that the higher the value, the more arthritis, the more severe the arthritis in the animal is. And then we compare the progression of the swelling of the joints as well as the redness and other clinical signs of disease. And you compare the gray, which is a control, meaning that's the animals that get really sick, and you compare them to the blue, the red, and the purple. The purple is a combination of specific drugs, and you can see that the combination performs better than the individual monotherapies. Now, there are other combinations, and that are shown at the bottom, where all the curves are overlapping with each other. I mean, the light blue, dark blue, meaning that these particular combinations do not afford therapeutic benefit above what their individual monotherapies do. Next slide. So the conclusion of that is that combinatorial therapy testing on a mouse model of RA, rheumatoid arthritis, reveals that there is a positive synergism between some, but not all monotherapies, right? So one should not expect that because 2 particular drugs work in isolation, when you combine them together, they would work better or work better. Next slide. This is an inflammatory bowel disease. There is 2 types of inflammatory bowel disease. And generally speaking, ulcerative colitis that affects the large gut, and then Crohn's disease that affects the distal part of the small gut. They're both, called inflammatory bowel diseases because they affect the bowel, but they are completely different diseases in reality. I mean, they're different mechanisms of disease and obviously different responses to treatment. And this is shown on the top, shows the percentage of body weight of the animal. So these animals have diarrhea, they have all the symptoms of colitis, and they lose weight as the animals get sicker, right? And so here you can see that the combination therapy in purple and in red and in and in red and in blue shows the monotherapies, and purple is the combination. You can see that the animals gain weight as opposed to what you see with the gray curve, where the animals lose weight and end up having very low weight. So that means that that in this particular case, the combination of these two drugs, ustekinumab and adalimumab, affords therapeutic benefit beyond what the individual monotherapies do. However, in the Crohn's disease model at the bottom, which is again, it affects the distal part of the small gut, you can see that the combination of these two drugs in purple on the far right actually does not work at all. And in fact, the addition of these two drugs together abrogates the therapeutic effect that one of the drugs has. And so, next slide. So what this is telling us that the same combinatorial approach can have a completely different therapeutic outcome on different types of inflammatory bowel disease. Again, Crohn's disease affecting the small gut versus ulcerative colitis affecting the large intestine. We have also established a contribution of genetic background on therapeutic outcome. What this means is that a drug can suppress the progression of Crohn's disease in one genetic background, in one type of mouse, for example, but not the same disease in a different genetic background, suggesting that the combination of these drugs might work in some patients, but not in others having the same disease. Next slide. So the general conclusions, and that's my last slide, these are some of the things that we've learned from these studies, is that overall, these preclinical studies provide a rationale for and support the clinical translation of a specific combinatorial therapies that may have superior therapeutic activity and or broader population coverage than the corresponding monotherapies in a specific immune-mediated diseases. Autoimmune disease. The data also indicate that the therapeutic effects of combinatorial therapies cannot be predicted on the basis of the effects of the corresponding monotherapies. Outcome is a function of disease type and genetic background. And the other lesson that is important from these studies is that in some cases, adding a second drug to a monotherapy that works can actually abrogate its therapeutic activity or even worsen disease outcome. So thank you very much.
Thank you very much, Pere. We continue with Dr. Ernest Choy, Professor and Head of Rheumatology Department at Cardiff University and leader of Work Package 8 Clinical Study Validation of Combinational Therapies.
Thank you for that kind introduction. In English, we have a saying called the proof of the pudding is in the eating. So my task in this work package is to look at all this wonderful recipe that has been cooked up by the different work packages in science and make it into a study that will test out which one is the most delicious for our patients to get treatment for that inflammatory arthritis. So that is the goal of this work package, is design a proof of concept one study that will allow us to test combination therapy in different diseases. And this is not a small task because we have many different diseases and we have many different combinations. And we have also predictive blood tests that may tell us whether or not the patient will respond to the treatment or not. But how can we do a single study that can answer all these questions? So to do that, we need a novel design, and we use two different technologies to do that. One is we're going to design a basket trial. So what we're going to do is to put each disease we want to test and each combination of treatment that we want to test into a basket. So this is a group of patients treated with a specific treatment. And we're going to follow this patient in each basket independently. That's number one. Number two is in order to make it most efficient, we need to do an adaptive design. Adaptive design said that it is a flexible design. So unlike standard clinical trials, where we have to recruit all the patients and look at all the response right at the end of the study to make a decision, we're going to design the study so that sometime during the stages of the study, we'll look at the observation we see and to make a decision on whether it is a combination worth us to continue on, or whether we are not seeing the expected result, and then we should stop that particular combination in those particular patients. And I'm going to use, in the DocTIS consortium, we're going to use a specific adaptive design, which is called Simon's two-stage design. And I will show you using the DocTIS clinical trial program to show you why this is called two-stage design. So on this slide, you will see two baskets that we intend to recruit, and we can include more. So there's a basket that includes patients with rheumatoid arthritis, and a basket that will include patients with psoriatic arthritis. It will be given a specific combination therapy. So the study will say that we will need something like 20 patients in order to decide whether the combination treatment is producing the desirable effect. But at the first stage, what we will do is to look at the results from 7 patients who have been fully studied. And we will say that from 7 patients, if we don't see any patient achieving the desirable clinical result, then the combination isn't going to achieve the likelihood of a big response. So it will tell us that we should not continue with that particular basket. But if we're seeing some response after treating 7 patients, we will continue as we have planned it and fully recruited 20 patients into that basket. So that at stage 2, that is the time when we would decide whether a combination would be achieving the desirable basket, because by then we will have recruited 20 patients. It will allow us to say whether the 20 patients response is meeting a very high level result that we anticipate. So one of our assumption at the beginning is that if combination therapy is going to be useful in our patients, and we should be expecting a much, much better response than we will expect from our standard treatment. And in this case, we expect combination therapy to reduce inflammation to nothing. So we expect the patient to get back to almost normality with combination therapy. So that's a very high level bar to reach, and we expected something like a third of the patient treated with combination therapy should be achieving this state of almost no disease at all. So that is a novel clinical trial design that we adopt in the DocTIS trial that will allow us to study multiple patient groups as well as multiple combinations in an effective and timely manner.
And thank you very much to all speakers for presenting such a complex project like DocTIS in a brief and clear way. I believe that your insights have truly helped us understand its key aspects, but if anyone has questions, please go ahead and write them in the chat as we now start the time for questions and answers. Don't see any questions? Nobody has written any question in the chat nor in the question. So if there are no questions, we will now conclude our webinar. Thank you all for joining us today. We hope you found this webinar and the DocTIS project interesting. If you have any further questions or feedback, please feel free to reach out. Have a wonderful day.